city. Dresden is beautiful city.
Yeah, it is.
I just come back last week from Dresden.
Was for business or for vacation?
For scientific visit to the University Applied Technique. I met Professor Catherine Hara.
So this has to do with radiochemistry? No.
Polymer radiation.
Polymers. They are strong. There's also an academic institute in Dresden dedicated to polymers.
Yeah. And I there under supervision of the Dr. Uwe Goh. Maybe you don't know also, yeah.
No, frankly speaking, these polymer sciences are far away from our small molecules.
Yeah. Okay. Have you visited Indonesia before?
No, never.
Okay.
So, well,
maybe if you have changed, we can invite you to be here in Indonesia.
I was planning to go to Hawaii. Hawaii is the place for the big conference on radiopharmaceutical sciences. Is it a connection between Hawaii and Indonesia? I think it's not so long distant.
What country?
From Hawaii.
Hawaii, yeah. Hawaii belong to America, yeah. But it's far away from Indonesia, in Pacific, I think so.
But it's from Hawaii to Indonesia is shorter distance compared to between Germany and Indonesia.
I don't know. It should be checked in the Google map.
So I was just thinking I am close to Indonesia when I'm kind of kidding. Yes, this is definitely something I really would like to do, to see your country.
Yeah. Because currently we have also scheme to invite professor from abroad to stay here, for example, for 1 month, yeah, and then to enhance our research group here, collaborate to writing paper, something like that.
Yeah, very good. This is a nice opportunity. Yeah, let's see. I'm ready. And it seems
okay, ladies and gentlemen. Okay, could you hear my voice clearly? Okay, ladies and gentlemen, thank you for joining our webinar. We will commence shortly. Assalamualaikum warahmatullahi wabarakatuh. As it is now late afternoon in Indonesia, I wish you everyone a pleasant afternoon and serenity. Welcome to the expert talks on radioisotopes, radiopharmaceuticals, and biodosimetry research and development held by Research Center for Radioisotopes, Radiopharmaceuticals, and Biodosimetry, National Research and Innovation Agency of Indonesia. Today, there will be a talk entitled Theranostic Fab Inhibitors, the Translation of Diagnostic Monomers to Therapeutic Dimers and Towards Instant Key Type Labeling at Ambient Temperature for Trivalent Radiometals. I am Ilma Derjatin. I'll serve as the webinar's host today. It is an honor to me to inform you that among us here, we have Distinguished Head of Research Center for Radioisotopes, Radiopharmaceuticals, and Biodosimetry Technology, National Research and Innovation Agency of Indonesia, Dr. Tita Puspitasari, MSc. Distinguished guest speaker, Professor Dr. Frank Roche, and respected moderator, Mr. Ahmad Kurniawan, DVM, MSc. I would like to apologize on behalf of Head of Research Organization for Nuclear Energy, National Research and Innovation Agency of Indonesia, Dr. Rohadi Awaludin, that he was unable to attend this webinar due to another important meeting. Ladies and gentlemen, please allow me first to read the agenda for today. The first agenda is the opening session. The second is a webinar session led by our moderator. Last but not least is the closing session. Also, please allow me to read the webinar's rule of conduct. To all attendees, please mute your Zoom meeting, as attendees are expected not to interrupt the speaker's presentation. If you have any questions, please type ask, then proceed with your name, followed by your institution and a brief question in the chat box. You may also ask questions by activating the raise hand feature on the Zoom meeting until the moderator invites you to. Due to the time constraints, the moderator will present only selected questions to the speaker during the discussion session. And if you wish to fill out the attendance list, please visit a link that will later be embedded in the chat box. Well, ladies and gentlemen, we are now in the opening session. Without further ado, I would like to invite on Dr. Tita Puspitasari, MSc, Head of Research Center for Radioisotopes, Radiopharmaceuticals, and Biodosimetry Technology, BRIN, for some welcoming and opening remarks. Ibu Tita Puspitasari, the time is now yours.
Okay, thank you, Mbak Ilma, as a MC for this event. Again, good afternoon, distinguished guest speaker from the Institute of Nuclear Chemistry, Johannes Gutenberg, University Mainz, Germany, Professor Frank Ross. Ladies and gentlemen, first of all, on behalf of the Chairman of the Research Organization for Nuclear Energy, Dr. Rohadi Awaludin, I welcome all of you to our webinar series, namely Expert Talk, in the field of radioisotope, radiopharmaceutical, and biodosimetry. As already mentioned in the flyer, our topic today is fibroblast activation protein theranostic and instant kit type labeling with radiometal. As we know, the need for radiopharmaceutical worldwide is increasing yearly, which has implication for the need to enhance research and development activities in this field. We do hope that by sharing knowledge, cooperation, and collaboration among us, like this webinar today, research activities in this radiopharmaceutical area can be accelerated. Last, today's expert talk can benefit all of us. I would also like to express my gratitude to the committee for preparing this webinar and to all participants for your willingness to take part in this event. Please enjoy talk and discussion. So thank you very much for all. Back to the MC.
Okay, we would like to express our gratitude to Dr. Tita Puspitasari, MSc, for the welcoming and opening remarks. Well, ladies and gentlemen, before we begin our main agenda, now we will be taking pictures together. So please, sir and madam, activate the camera or video function on your Zoom application. I'd like to invite the IT team to assist in taking pictures. Please be prepared because I'm going to count from 1 to 3. Okay, again, I would like to remind you to please turn on the camera video on your Zoom meeting. Okay, ready? 1, 2, 3. Please wait a moment and hold in your pose while our IT teams are taking our pictures. Okay, I think we've finished taking pictures. Thank you for your cooperation. Well, ladies and gentlemen, we have now reached to our main agenda, the webinar session. All presentation and discussion will be presided over by Mr. Ahmad Kurniawan, DVM, MSc, as a moderator. Allow me to read his brief biography. Mr. Ahmad Kurniawan, DVM, MSc, is a junior researcher at Research Center for Radioisotopes, Radiopharmaceuticals, and Biodosimetry Technology, BRIN. He has extensive experience in drug discovery and radiopharmaceuticals development for cancer diagnosis and therapy, especially in mammalian cell culture for in vitro testing of radiopharmaceuticals, laboratory animal science, preclinical molecular imaging, and biomolecular analysis. After completing his undergraduate studies in the field of veterinary science and veterinary degree at Erlangga University, Indonesia, he continued his master's studies in biomedical sciences at University of Glasgow, United Kingdom in 2021. So, ladies and gentlemen, let's welcome our moderator, Mr. Ahmad Kurniawan.
Yep. Thank you, Ms. Ilma as host, and good afternoon, everyone. Good morning, Professor Rose in Germany. So it's been an honor for us to have you today to give sharing session for all the participants, all the audience. And to all the audience today, Professor Rose will give two topics. The first is FAP inhibitors. We know that the fibroblast activation protein, or FAP, is has important role in tumor growth and other disease involving tissue remodeling. And it has become a very interesting target for the theranostic radiopharmaceuticals. And the second one is about the instant kit radio labeling using radiometal. So before we continue, I want to read a short profile of Professor Rose. Professor Rose was appointed a full professor at the Institute of Nuclear Chemistry, Johannes Gutenberg University in Mainz, since 1996, and he focused on the production of carrier-free lutetium-177 using a reactor and the establishment of cation-based germanium-68 and gallium-68 radionuclide generator. On the other hand, Professor Rose also conduct a research cover the whole radiopharmaceuticals molecule, including new class of bifunctional chelators and instant kit type production of radiometal. And last but not least, I want to congratulate Professor Rose sophisticated achievement in last year, which obtained Havasi Medal Award. And Havasi Medal Award is a premier international award of excellence honoring outstanding achievement in radioanalytical and nuclear chemistry. So the presentation will be around 45 minutes. And as we discussed before with Professor Rose, the first is about FAP inhibitors. And after that, if Professor Rose want to have a 5-minute break, so we can continue to the second topic. And the discussion session will be after presentation. I think it's approximately 1 hour. So hopefully all the audience can put the question on the chat box, or you can raise your hand after the presentation. And Professor Rose, time is yours.
Yeah, thank you very much for the nice words. I always remember very well when you visited the institute at my university with your colleagues, and I think it was very nice. We had a nice time together. So now, and thank you, of course, for inviting me to this seminar. As mentioned before, and I will give two different presentations on two different topics. While maybe they have some relationship, I will discuss this in the second one. But here I would like to start with, as you mentioned, a very important topic in radiopharmaceutical sciences and cancer diagnosis and treatment, which is based on a very interesting molecular target, the fibroblast activation proteins. My lecture will start with some basics on the molecular target and the radiopharmaceutical or the molecular targeting vector. And then I will come to the medicinal chemistry part on how to develop radiopharmaceuticals. And from the success of the diagnostic tracers, I will finally come to the recent developments for therapeutic analogs. So that's basically the first part. What is cancer-associated fibroblast and medicinal chemistry? The fibroblast activation protein is a component of the tumor microenvironment or tumor stroma. And this tumor microenvironment represents a large amount of the total mass of a tumor. And what is interesting for our field in radiopharmaceutical chemistry is that there is a certain molecule, a protein, the fibroblast activation protein, which is overexpressed in those cancer-associated fibroblasts that have a different role, and this is still under development. It's very important for many diseases. including fibrosis, but here we will focus on those cancer-associated fibroblasts. The FAP, the fibroblast activation protein, as I mentioned, is a huge protein, a protease, which is the basic component of these cancer-associated fibroblasts. The protein itself, in its structure, is a dimer consisting of two identical parts of the protein. According to their functionality, this is a proline selective peptidase. So it means that the protein change is cleaved by the enzyme at those protein positions within the huge amino acid sequence of that protein. There are a number of similar proteases. They all belong to these DPP4 family, proline peptidase family. And this is important for the molecular chemistry or the development of radiopharmaceuticals, because we will always have the challenge to develop small molecules which are very selective for fibroblast activation protein compared to the other members of the same proline selective protease family. The development of the most important FAP inhibitor goes back to research and development at the University of Antwerp in Belgium. And this basically had nothing to do with radiochemistry or radiopharmacy. It was entirely a project of pharmacy, in particular of a group of medicinal chemistry. The head of that group is Professor Peter van der Weken, and his team started to developing this small structure, which is the initial lead compound for the FAP. And they always, following the synthesis of the compound, measure the affinity, binding affinity in protein assays to the FAP, which is our important target, but also to the other members of this protease family. And you see here that the first that compound had a nanomolar affinities. This is micromolar, but this is, I would say, sub-micromolar. It's 110 nanomole binding affinity, while the selectivity already was quite good compared to the other members of the protease family. Then they learned a lot. That's what medicinal chemists do from these first lead compounds, and modified the basic structure of this small molecule, introducing, for example, here nitrogen in the heterocycle. And this new derivative now improved binding affinity to the FAP by 1 order of magnitude and still retained high selectivity. However, the final molecule they come up was further modified, and they introduced this difluoro substitution in the proline sequence. And that particular molecule now has a very high binding affinity of 3 nanomolar. And the other members are still micromolar affinity. So this now represents the most important small molecule FAP inhibitor, characterized by both high binding affinity to our target and selectivity. They published this basic research some years ago, and as I mentioned, there was no relationship to radiopharmacy or serognostics at all. Interestingly, they used the molecule to develop new drugs for fibrosis, but this was already studied in clinical phase trials, but did not show superiority compared to other drugs known as anti-fibrosis agents. So that was basically the end of the story of the medicinal chemistry for this nice molecule. However, the next step of the development was the translation of this inhibitor molecule to radiopharmaceuticals. And here the basic strategy is almost in all cases to stay within the molecular structure, which is called UAMC 1110, which means University of Antwerp Medicinal Chemistry. That's the group. And the question is how to translate this inhibitor into a radiopharmaceutical. This is what we know from PSMA inhibitor-based radiopharmaceuticals or the octreotides for neuroendocrine tumors. So we need to add a chelator structure if it is about radiolabeling with radiometals. The preferred position in the quinoline member of this inhibitor is shown here because modifications here retain high affinity and selectivity of the inhibitor. The first to understand that, to realize that this inhibitor could be part of a very important class of radiopharmaceuticals was again a group of Martin Pomper at Johns Hopkins, and they filed a patent 3 years ago, 2019, claiming the first radiopharmaceutical structure of FAB inhibitors. So you see here the inhibitor part of the molecule, a linker moiety, and they used DOTA or DOTAGA as a chelator in their patent application. Actually, they never continued with human applications, and both for the this derivative and the D-fluoro version. But this is the first step to go to radiopharmaceuticals. So the basic credit for first patient application goes to the German Cancer Research Center in Heidelberg. They stay within the concept, having the inhibitor structure here in that gray box, having the chelator here is a DOTA instead of the DOTAGA, it doesn't matter so much. But what they introduced here was a nitrogen heterocyclic moiety in the linker motif of the compound. We at Mainz did a similar approach. We again remain the high affinity and selectivity inhibitor, but introduce a squaric acid motif, which is a nice chemistry for asymmetric conjugation of two molecules. And we varied the chelators. We also use DOTA, DOTAGA, and DATA. This is actually the DATA structure I will discuss in the second presentation. So here now we are back to the basic structure of a radiopharmaceutical labeled with radiometal, which contains of a targeting vector shown here in red, linking chemistry, coupling chemistry, spacer chemistry, and a covalently attached chelator to coordinate radiometals, basically trivalent radiometal. This concept was extremely successful, and the Heidelberg group indeed was a pioneer in doing that. And they published soon the first PET images they demonstrate that there is uptake of the gallium-labeled FAPI04, and then later on a number of modifications here, basically in that part of the molecule, and could show that there is interesting diagnostic potential for a variety of cancer. And saying this, I would underline the main feature of the FAP inhibitor family. They target tumor microenvironment, tumor stroma. And basically, it doesn't matter what tumor it is. The binding to tumor microenvironment is tumor unspecific. It's like FDG in imaging tumors with PET-CT. It is an unspecific biochemistry. And the same is true here for these FAP inhibitors. It's not an unfortunate, it's great progress that now we do have a pan-tumor imaging tracer. In our case, we did very similar developments with our squaric acid motif compounds like DOTA squaric acid FABI or DATA squaric acid FABI have been developed. The advantage here of the DATA, and I come back to that in the second lecture, is that labeling is possible at room temperature with instant kit type chemistry. After 1 minute, it's close to 100% radiochemical yield compared to the DOTA chelator, of course, which requires higher temperatures. So this is what I will discuss later on. We always, as mentioned earlier, measured the binding affinity of our molecules to the FAP itself and to the most critical other enzyme, PrEP. And you see that our molecules without the chelator like DOTA, without the radiometal like DOTA, squaric acid FAPI, and the DOTA squaric acid coordinated with gallium, they remain the nanomolar binding affinity. And you see here the high selectivity in micromolar. And we are pretty much the same in terms of binding affinity as the real inhibitor molecule of the Antwerp group. This was our first small animal PET imaging with 3 mice with HT29 xenograft mouse model. And this here is the bladder. But you see here nice accumulation in the tumor xenograft and low background in the remaining part of the animal. So it was very promising. So our collaboration partners in India at the All Indian Institute of Medicine, Medical Sciences, they used these compounds immediately for PET-CT imaging of, again, different kinds of cancer. And this is these are images for breast cancer patient over time. It is clear visualization of the FAP inhibitor already about 20 minutes post-injection. This is 1 hour image, 2 hour, and 3 hour images. This is lung cancer, and so on. So we basically see what also the Heidelberg group reported. There's a good retention in the variety of tumors. It's a fast uptake, but you also see that after 3 hours, slowly the activity is released from the tumor stroma. If you are interested in a kind of a summary of all these developments, we recently published a review article in the journal Radiopharmaceuticals to cover all these aspects I just mentioned. The basic message clearly is that FAP inhibitor breast targeting works. So here we are now. Let's say this is a tumor microenvironment, a tumor stroma. And now the gallium-68 labeled FAP inhibitors really show up in these tumors. And we can clearly say this is now a pan-tumor imaging agent. There are studies undergoing comparing the FAP inhibitor with FDG, and in some very important kinds of cancer, it seems that these FAP inhibitor-based radiopharmaceuticals are even superior compared to FDG imaging. Yet this is the first step, and as always in diagnostics, we expect that we will sooner or later have a therapeutic option of those molecules by just substituting a therapeutic trivalent radiometal for the gallium-68, for example. There are great expectations also in terms of commercialization of this group of radiopharmaceuticals. But there's a problem. The structures of the, let's say, radiopharmaceuticals shown here again, applied to the FAP inhibitor, show a very short biological half-life. For the FAP inhibitor 04, the biological half-life is about 2 hours. For FAP 46, this is 8 hours. But for the two therapeutic radiometals, tritium-90 and lutetium-177, if with their corresponding half-life, if you calculate the effective half-life, you come up with those numbers, 8, 14 hours. And this definitely is not long enough to deposit a sufficient radiation dose to the tumor. Also, many groups tried to label this kind of structure with tritium-90 or lutetium-177 or actinium-225. They see some effects in the patients, but this is not really as most of us expect. But there is a problem. And what is the challenge? The challenge is clearly, it's obvious, we must prolong the residence time of these FAP inhibitor-based radiopharmaceuticals in the tumor stroma, without, of course, adding radiation dose to healthy tissue. There are different strategies common in medicinal chemistry or radiopharmaceutical sciences is to modify the linker or spacer. Basically, this is what the Heidelberg group did without substantial progress. We may introduce albumin binding structures, and currently this is a strategy many people do, also industry. But we should figure out later whether this is really addressing this gray point here. Do we accumulate in healthy tissue for longer? Peptides are used instead of inhibitors, FAP specific peptides. This is also a commercial pipeline. But what we tried is to develop FAP dimers. And in the last part of my presentation, I will focus on the chemistry of that, just to illustrate the basic concept. Compared to a monomer, I would say all these structures I have discussed until now are monomers. A dimer looks like that. So we have two identical FAP inhibitor moieties coupled to one chelator part. Our first approach was this, I would say, linear construct with a macrocyclic chelator coupled with two of the same inhibitors. Chemistry here, one of the molecules basically looks like that. We have a DOTAGA-like structure, and we couple the FAP inhibitor using our squaric acid motif to the chelator. The in vitro chemistry works nicely. This is a FAP affinity of the dimers with a DOTAR and the DOTAGA. And you see we still keep the high binding affinity and the selectivity for those compounds. So what we next did is based on these convincing in vitro data to show how do they perform in patients. And this is an example, first patient where we compared the monomer with the dimer. This is a 60-minute image, and this is a 60-minute image for the dimer. We call it a homodimer because they are exactly the same structures. And we see high uptake in the tumor, but we still also see high blood pool activity. This is a 3-hour image, and compared to the image of the monomer obtained for 3 hours, we see further increased activity in the tumor. But still, the gallium compound stays in healthy organs. This was confirming our concept of the longer retention time, but would not produce a better diagnostic agent. This is different for the lutetium-labeled compounds. This is our monomer, which is then taken up in the tumor, but is released over time. Fast release as for all the compounds we have seen also from the Heidelberg group. But this is the case here now for our DOTAGA dimer. This is a patient we studied. This is a radiation-resistant thyroid cancer patient, FDG image, 131 iodine images. And this is the PET-CT of the patient with our gallium DOTA squaric acid Fabry monomer. So we see a lot of tumor mass in this patient. And now here is the retention of the lutetium-labeled analog. Let's just, for clarity, focus on this large tumor mass. There are different planar images, 6 hours, 1 day, 2 days, 4 days, a week after injection. You see still the lutetium-labeled dimer remains in the tumor. And this is what we expected. We see what we wanted to see. There's a long retention over days compared to a couple of hours for the monomers in the case of the dimers. This is now confirmed by a number of other patients. You see always a similar case here. That's a patient again with medullary thyroid cancer. This is data NOC for somatostatin expression, the FDG, and the gallium-labeled Fab inhibitor monomer. And we reproduce the accumulation pattern we see in the gallium PET-CT with monomer in the lutetium dimer imaging. This is 2 days post-injection. So now we can expect that this long retention will deposit, hopefully, enough radiation dose to the patient to treat the patient. And this is now being done systematically in New Delhi at the group of Professor Baal. And we always reproduce these long retention time you see here, 4 days post-injection. And then what about the clinical success of this application? This is another patient. This is a gallium PET-CT monomer. Then this lutetium distribution over days, still there after a week. And this is a success. You see now in a post-therapy PET-CT image, how the tumor mass is reduced. This is before the Fabry dimer therapy. This is after it. You can quantify that in terms of lower size, lower SUV, but also in terms of clinical parameters, in terms of quality of life. And this is great success. And this is, I just showed you 2 more examples. A similar story now compared to what we know from neuroendocrine cancer patients with the somatostatin target, and compared to the prostate cancer with PSMA as a target. We see decrease in expression of the tumor, we see decrease in SUV, we see improvement in all parameters of the patient. Well, just another example. This was nicely summarized in the cover story of the journal Thyroid, with, I think, 15 patients included there, demonstrating that the whole body half-life is 88 hours. The absorbed dose per cycle, the decline in thyroglobulin levels significantly. And what is important, none of these patients experienced serious, significant kinds of toxicity. The group also continues now, and they have now submitted an abstract to the SNM meeting in Chicago next year, demonstrating Kaplan-Meier curves with progression-free survival and so on. Yeah, they also did dosimetry, and you see that the dose, radiation dose to the tumor is extremely high, while the radiation dose, the mean absorbed dose, is relatively low for healthy organs. The critical organ is the colon, but this is acceptable. Let's compare that with the mean absorbed dose for the PSMA compounds, either 617 or INT. We see that the dose delivered to the tumor is much higher compared to the PSMA inhibitor-based therapeutic compounds. But this is tolerated absorbed dose limits, and you see the liver is not a critical organ at all. So this is all very promising. I should mention that the basic work was published for thyroid cancer patients, but because the target is a pan-tumor target, the cancer-associated fibroblast, although the radiotherapeutics may act as a pan-tumor therapeutic. And here's an example for breast cancer. The uptake is also long, and this is demonstrated for prostate cancer, neuroendocrine cancer. And this is really where we are right now. And for the last 2 minutes, let's go back to chemistry, kind of outlook. The next generation was compared to the monomers, what we proposed with the homodimers in this kind of molecular design. This was adapted by other groups. They stay within the homodimer concept, but position the chelator in an exo position right here. And here's a number of molecules behind this structure with the two identical inhibitors and the chelator in this exo position. We call it a tris linker strategy. Well, you may think changing from dimeric to multi-dimeric or compounds, something like that. Just following our concept that as more inhibitors you have in one molecule, as longer the retention in the tumor microenvironment would be. But it's not just fancy, it's not just fantasy. There are already experimental studies with this or similar kinds of these multimers. Another option would be to change the homodimer concept, either this geometry or this, to what I call heterodimers. Let's have the FAB inhibitor there as one vector to the tumor microenvironment with the CAFs, but add another different vector, which adds in terms of binding or retention. And indeed, those studies on hybrid compounds are going on with combining different vectors. And recently there was indeed a first paper on such a heterodimer with PSMA as one of the vectors and FAB inhibitor as the other one. My last comment would be on F18 labeled compounds. You know the story that also for the prostate cancer theranostics, there's work on F18 labeled analogs because of the availability and the advantages in commercial distribution of F18 labeled compounds versus gallium-68 labeled compounds. And this is the same thinking now for the FAB inhibitors. The strategy would be to remove the chelator, remove the linker chemistry, and simply add the radiolabel directly on the structure of the, I would say, Antwerp inhibitor. And this is currently under development. I know there is already great success in chemistry and preclinical evaluation. So I could imagine that in a couple of years, we may have a commercial availability of both FDG and F18 labeled FAB inhibitors to complement each other in the diagnostic evaluation of a variety of tumors. So with this, I would like to conclude here. This is my group, and here also Moon was a PhD student who was basically working on the dimer strategy. So this is our collaborator group in India. It's Professor Bal and two nice young people over there. So thank you for your attention for this.
Thank you, Professor. And do you need, I think you need a break around?
No, I just changed the...
yeah, no worries. So currently we have 60 participants. We expect around 30, but we have 60 here. This will come from university, hospitals, and from our institute. And I think we have a researcher that focus on the biodosimetry. And to all the audience, if you have any question, you can put that on the chat box and we will discuss it later on. Or after this, you can, after the second presentation, you can raise your hand using the Zoom features, and we will select the participant for the direct question. So we still have the second, this is a really interesting, Professor, for your first presentation, I think. The first time I come to Mainz, and there is still under development for the FAB inhibitor now is goes to clinical trial with a very promising result, I think.
You know, I absolutely agree. As I mentioned, that it's I retired last year, and it's a kind of pity that I have to retire in these exciting times of new developments.
Yeah. Especially, and I'm really interested to see that when they finish the clinical trial, I think that is will be in India, right?
They continue with patient applications, and they are absolutely excited. We can discuss this later in our discussion here.
Yeah.
Well, this is now the second. Do you see my PowerPoint?
Yeah, that's perfect. You can start.
Okay. Then this is a topic I also like, and you have seen that when you visited us, that our group spent a number of projects and PhD thesis on this research on interesting chelators, which make the life of us in the daily preparation of radiopharmaceuticals easier. So this was kind of the motivation. Well, Okay, let's start here with the motivation again. So what I wanted to address in this research is that this here on the left-hand side again is the basic structure of theranostic compounds, with a targeting vector, which could be a PSMA inhibitor or the somatostatin, the octreotide peptide, linker chemistry, and then the chelator attached to it. We learned that DOTA and DOTA derivatives are extremely convenient to our field. But they need elevated temperature, 95 centigrade for labeling. I would say it's not, it works, of course. There's no doubt that it works. But on the other hand, the majority of radio metal labeling in nuclear medicine is being done with technetium-99m. And here the labeling protocol is very easy. It consists of just eluting the 99 molybdenum technetium generator into a lyophilized vial, shake it, and that's it. And then inject it. This is the state of the art of labeling for SPECT imaging. Very easy, very, I would say, low legal regulatory issues, easy to do. And this was really one of the reasons that the technetium-99m labeled compounds so successfully went into clinical application, because everybody could do it. Just elute the generator into a lyophilized vial. Don't worry, it works. So, and then that was my point. Could we achieve something similar for gallium-68 labeled analogs? And this, of course, would need a change in the chelator structure, not further working on the DOTA, substituting a new one, or having something else stay with the DOTA, but do something that the radio metal can easily, more smoothly fit into the cavity of this macrocyclic chelator. So I will just describe what we did here for the first attempt, always keeping in mind that we would like to have labeling achieved quantitatively at room temperature. So the first point, strategy we tried was to modify the solvents in the labeling solution. And this goes back to our basic publication on the purification process, the post-elution purification of the generator itself. And here we used a mixture of hydrochloric acid and acetone, which was the basic chemistry of this purification process. But what we always learned is that using this post-processing protocol, the labeling of the radiopharmaceutical was always very elegant and fast and quantitative, while the others not using this post-processing strategy reported lower yields. They needed to heat the solution for longer, and so on. And then that brought me to the idea that there seems to be an effect of the acetone in this solution. So we now did a little study on comparing for a proof of principle compound, DOTA-NOC, with gallium-68, the effect of the additional solvent. Just to have the basic reference, if we label DOTA-NOC at close to 100 centigrade, this is how the labeling proceeds, depending on the labeling time. If we reduce the temperature down to 70 centigrade, already it's much less. So this is the basic fact. If we work in pure aqueous solution. Low yields at lower temperature at short time. But if we now add to the same solution different non-aqueous solvents like acetonitrile, ethanol, isopropanol, to the same solution, same temperature of, let's say, 70 centigrade, then this is what we have seen for pure aqueous solution. But in contrast, with a content of 30 volume percent of that non-aqueous solvents, you see a dramatic increase in the labeling. Absolute yields, in particular also in the speed of the labeling, it's much faster. So there's a significant higher labeling yield for a given action period. The effect is a little bit depending on what non-aqueous solvent you may use, but tendency is always the same. There is a clear effect of the ethanol. Ethanol, for example, if you do the labeling with 30 volume percent of the ethanol, you double the yield, or in particular after 5 minutes, it is really a dramatic effect for the labeling yield. And this can also be observed for, depending on the concentration of the ligand, we achieve higher yields at lower precursor concentration. And this is a dramatic effect for the gallium labeling. Actually, it also is true for lutetium labeling of DOTA conjugated molecular imaging vectors. Again, the same tendency. And the reason for that, I believe, is the following. The radiometal is, like all metal ions in aqueous solution, surrounded by a strong sphere of water molecules, hydration spheres. There are classifications in the first and the second and the third hydration sphere, and depending on the charge and the radius of the metal ion, those molecule waters are really attached very strongly to the central ion. But if you want now to see a complex formation, this hydration sphere is kind of a shield, and it prevents the access between the central metal ion and the donor atoms of your chelator. So adding non-aqueous solvent molecules will destroy the hydration sphere and weaken the interaction. So you remove a bit kind of this shielding effect of the water molecules. And that's, in my hands, the reason that we see that such a dramatic effect of adding non-aqueous solvents, and then the complex formation is easier and more efficient. This was published in a very nice paper with coordination chemists to quantify all these effects. Well, it works. You can, if you have some trouble, I would always recommend you to add some drops of ethanol and see what happens. And I can promise it will always be helpful. But, you know, there are different, I would say, also very important applications of ethanol. So different to decide what's the best. So this is kind of something you may do. But the better approach is, or maybe, let's say, a parallel approach, because it always will work in the presence of ethanol, is to think about the modification of the chelator. And a new chelator would allow complex formation at a lower temperature compared to what we know for the DOTA tetraazar macrocycle. And this takes us back to the history of radiopharmaceuticals, radiometal-based radiopharmaceuticals. We always had, in the beginning, acyclic chelators like DTPA. Only later on, the DOTA was introduced. It was a kind of present handed over from our lanthanide chemists, which who are working in lanthanide chemistry or MRI imaging, contrast agents. They learn that it's better to use a macrocycle instead of a acyclic chelator for trivalent metals like gadolinium. That was adapted by radiopharmacy, and we know now that it works. Is the disadvantage that you may need higher temperature. Labeling with acyclic structures like DTPA or DFA is always working very smoothly at room temperature, but stability in aqueous systems or in biological environment is a bit problematic. And that was the philosophy behind the change from acyclic to macrocyclic structures. Maybe NOTA is an exception because NOTA perfectly fits to the gallium carrier. But this is a strategy to have the advantage in easy labeling at room temperature for the acyclic structures and high in vivo stability for the macrocyclic options. Well, there are new developments. I would say in the last decade, there was a development of new chelators, acyclic challengers for the gallium labeling. And I think HBAT is a prominent example, which is the chelator used for the PSMA 11 molecule. This can be labeled at room temperature. This is also a molecule reported to allow labeling at room temperature, and the same is true for the THP versions coming from a group in London. Our contribution was the data structure, which we believe is a hybrid structure. Why hybrid? Hybrid says that we remain a cyclic part in the molecule, which is responsible for high in vivo stability, but also have an exonitrogen, which creates an acyclic moiety, which is responsible for labeling at room temperature. But this is what the reason we call this is a hybrid chelator, and indeed it works. So we did initial labeling of this chelator, and it works at room temperature immediately. You know, this is 100%, this is 90% here. We are at 98 or 97 or 99% immediately. Well, this is true for the basic chelator. So we later on spent a lot of chemistry to develop bifunctional chelators based on this chelator data, asking what is the best substitution for covalent coupling to a molecule, either here or here, and we came up with the substitutions at the nitrogen. So the best molecule in our hands, the best bifunctional chelator is that one here, and we refer to it as a data 5M. M means that we leave this methyl substitution at the exonitrogen position, and we add here at the 6th position 5 carbon atoms to a carboxyl function for a covalent amide or other coupling to a molecule, data 5M. Well, this was investigated in terms of thermodynamic stability and complex formation constants. It works. And now we do have an alternative compared to DOTA, but what we really know from literature is we must be careful. It's not enough just to change chelator. We must also, and see how thermodynamics and kinetics work. We must make sure that the pharmacology is also good enough for human applications. So there was a great paper from Helmut Mecker's group saying that the chelate makes a difference, not only in terms of labeling, but also in terms of pharmacology. So that was the reason that we not only discussed gallium labeling or copper-64 labeling, we wanted to see what is the pharmacology of a real compound. The basic proof of principle study we did was to compare then DOTA talk with data 5M talk. Same vector, the octreotide analog, same coupling chemistry as at the N terminus of the peptide, amide formation to the chelator. Actually, the same basically here is shown here with our data 5M. So this was a complete analog. Now we first saw reproduce that labeling of this gallium data talk is extremely efficient, and after 1 minute already, depending on the post purification of the generator, it gives you labeling yields above 95%, which is basically needed for direct application of the labeling solution. And this is what we wanted to see. We now have approached the situation similar to kit labeling of technetium compounds. So this is now how it works. We do have the generator post-processing and have in that while the lyophilized product. And we do not need to wait. We do not need to heat it. It gives us immediately a radiochemically and radionuclidically pure gallium radiopharmaceutical ready for application. But now back to the pharmacology. Well, we see a difference in the binding affinity of these octreotide derivatives to the somatostatin subtype 2 receptors in cell studies. We remain high binding affinity, but it's a bit less than what we have seen for the DOTA analog. But in the tumor animals, we see same performance, same uptake, same specificity. We can block this for the DOTA talk, and we can block that for the data talk. We see organ distribution is pretty much the same. We see less uptake actually in the kidneys and a bit less uptake in the tumors, but the difference between tumor uptake and the uptake in healthy organs is in favor of the data talk analog, and that's what you see here in one and the same patient. This is data talk compared to DOTA talk. You see here less uptake in the liver. So then the ratio between the SUVs in tumor and liver is indeed in favor of the data talk. So we have, let's say, double the contrast with the data talk. Well, this was reproduced by various groups in the world, and again, there have been nice papers on both the in vitro evaluation of this compound and the clinical performance, a very systematic comparison of the data talk compared to the DOTA NOC in neuroendocrine cancer patients, and concluding that the imaging quality is superior in case of the data talk. An interesting story here is what about the neuroendocrine cancer tracers if it comes to antagonists. These TOC or NOC, these are agonist cancers. And we recently published a study with the antagonist version, which is our data LM4. This is a substitution in the peptide sequence. Now this is data 5M added again. And again, preclinical evaluation demonstrated that the compound works. There's a nice, clear evidence that the data antagonist is even superior PET tracer in terms of affinity and in terms of cell binding, in terms of organ distribution in animals. And it also now works in patients. You may know that what is the disaster in neuroendocrine tumor imaging with the agonist, the octreotide derivatives. After a while, in particular, if patients were treated with lutetium or actinium-labeled agonists, octreotides, the patient in many cases does not demonstrate further expression of the agonist binding sites. So still there's neuroendocrine tumor mass, but the agonist tracers do not bind anymore. And now here comes the option for the antagonists. Antagonists are believed to have about 10-fold higher expression compared to the agonist binding sites of the somatostatin receptor. And indeed, this is what we see here. This is one of those patients. This is with a Tate tracer. You don't see so much tumor mass, but if you use the gallium-labeled antagonist, you really see this great tumor mass still present in the tumor. Well, this is our chemistry behind the FAP inhibitors. Another example where we believe that the ease of labeling could be an advantage also for the imaging of tumor microenvironment with the FAP inhibitors in targeting cancer-associated fibroblasts. This is something I have reported earlier. You see that in vitro data compounds perform in a similar way compared to the DOTA analogs. And also in human applications, the data 5M really works as good as the DOTA versions. This is published and this work is really going on. There's clinical useful application of these data conjugates. This is now not about the FAP imaging, or this is nice, as I mentioned earlier, but it works. The point is, again, that we may have and very easy to prepare analog of a gallium-labeled FAP inhibitor available. So obviously with the data structure and gallium-68, this is a success story. But in the last minutes, I wanted to come to the therapeutic analogs. So do we also have this concept available to instant kit type labeling for therapeutic isotopes? Because the data is only working exclusively for gallium-68. So yes, the answer is yes, but we need to have an extension of our basic chelator structure by just adding one carboxylic moiety more to the data. Here at that nitrogen, we do see only one carboxyl group. That's fine for gallium labeling, but for labeling lutetium, we need more donors. And ADSTA, that's the name of this structure, is really the structure which works for lutetium. It labels at room temperature. This was shown with PSMA 617, comparing the labeling with gallium, scandium, and lutetium at room temperature. See how labeling yields, and well, chemistry and biochemistry seems to work. And now we are thinking really to use ADSTA labeled structures for therapy. Let's come back to this one patient I explained a minute ago for the antagonist binding sites of the neuroendocrine cancer. This is a patient which demonstrates high uptake with the antagonist, but not with the agonist. So how will you treat now this patient? The lutetium-labeled agonist will not bind. We would need to have a lutetium-labeled antagonist. And this is the need for an ADSTA LM4 to treat this particular patient. And exactly this is what we have demonstrated recently. I cannot have not time to show you that in how that works, but it works. And just a final remark: this ASTA5 only also works with indium 111. So there's a number now of clinically relevant tracers ready for clinical use, which can be labeled in instant kit type fashion, and the extra option is to do it in a similar way for lutetium and indium trivalent radiokaryons. So with this, I would like to list all these many collaborators we have in this long-term development, and thank you for your attention.
Great. Thank you very much, Professor, for your amazing presentation. And I think, wow, there's a really nice development, especially when we talk about the recent theranostic agent, how we can have the theranostic agent with the high in vivo stability, and we can do the labeling in the room temperature with a lot of modification for the structure. So, so we have a couple of questions here, Professor. I tried to pick one question from the chat box. I think it's a really relevant with our current situation in Indonesia, based on your first topic. So it's from Mr. Basit Fabian. I think you remember him. He come with me to Mainz. He asked about the targeted therapy when we developed the targeted therapy like lutetium-177 FAPI or PSMA. So is it possible to use a neutron activated therapeutic radionuclide? It means the lutetium-177, which has a lower specific activity compared to non-carrier added radionuclides. Maybe you can give a response.
The answer is yes. Yes, you can. And some of the first patient studies in New Delhi at AIIMS, they used the reactor-based lutetium-177 because the situation is similar to Indonesia. They do not currently have their own carrier-free lutetium-177 production available. So they started with this compound, and clinically it works, I would say, exactly the same way as the carrier-free version. And there's a good reason for this, because it is different to what we know from prostate cancer therapies. Here, we believe it makes a difference because of the specific activity. With a lower specific activity, the total amount of the PSMA radiopharmaceutical is higher, and then there's a concern that this saturates the PSMA at the cell surface of the tumors. And this is true because there is a saturation problem for this PSMA target, the same for the somatostatin target. But this is not the situation for tumor stroma. We know that we still have to learn a lot. But from what I reported in my first lecture, from the developments in Antwerp, where the first inhibitor was used in clinical trials for fibrotic diseases, they have the impression that there is not so much saturation danger, I would say, in targeting the FAP tumor microenvironment. It would definitely, I would say, need a very systematic comparison of the two preparations. But my impression for what I have seen is that there's not, or I say it another way around. It is no problem to use reactor-produced lutetium-177.
All right, great. So basically, we can use the reactor-based lutetium-177 for yeah. And so because we have a lot of participants here, and not all of the participants is has a radiochemistry background. So if there is any participant want to ask the question directly, you can raise your hand using the features on the Zoom, and I'll try go to the next question. I think it's from Mr. Amal, is talking about how about the stability aspect? In the first presentation, you talk about the 5 inhibitor. Which one is more stable? Is monomers, homodimers, or heterodimers?
Well, we have measured stability for all the compounds, and they are all stable because the stability is related to the chelator chemistry itself. And then this is something we do not touch. So we measured traditional stability in phosphate buffer and in human serum, and it remains stable for a sufficient period of time.
All right, great. So I'm just curious, Professor, for your first presentation, what do you think about the recent trend of therapeutic radionuclides, let's say like actinium-225, when we do the labeling for 5 inhibitor, or the like a bismuth? Do you think that will give a promising results for the tumor microenvironment?
Yeah, this is a very good question, because again, here we still have to learn a lot about what's going on in the tumor microenvironment. There are two extreme positions. You could first have the point of view that you have the accumulation in the tumor microenvironment. This is like a shell around the tumor, like say, let's take the example of the orange. We have a lot of oranges in Indonesia, right? And when let's say what our molecular target is just in the shell. Do you say it's a shell in the orange?
Yeah.
It's I use the word shell for the nut, but I'm not sure for the orange. But you know what I mean.
I can imagine that. Definitely.
That's where our target is. That's where the cancer-associated fibroblast is concentrated. It is not inside the orange fruit. It's in the surrounding shell. Now, if you accumulate your therapeutic tracer only in that shell, and this is a different situation compared to prostate cancer or neuroendocrine cancer imaging, you stay with your tracer in the shell. Then you could argue that it is good to have a longer range of your therapeutic particles, which also penetrate the tumor mass inside, so the orange fruit. And then you would need, let's say, yttrium-90. This would have a long range inside the tumor and affect both the stroma and the tumor same time. Or you could argue the other way around. You could say, I prefer to exclusively, but to a higher degree, destroy the stroma only. And if your therapeutic strategy is to focus on the stroma, then a short range therapeutic imager would make sense, like actinium-225. But and this is true. And we try both options, but we have to learn a lot. But because these are two different strategies, and the basic question is it really sufficient to destroy the stroma? Because when destroying the stroma, according to the functionality of the tumor stroma to provide the tumor with everything the tumor needs to progress, then you would expect that the tumor will die. By just destroying the stroma and all the connections to the blood stream, you expect that the tumor will have no further supply and will shrink accordingly. So these are the two strategies, and I believe they both make sense. But what we see, but we do not have enough data. So with lutetium, I would say we are on the safe side, and we see that it works. But I would believe it is worse to in a clinical trial to verify the effect of actinium. Frankly speaking, the group in India treated a number of patients with actinium-225, is our dimer compound. And what they observe from the patient response is very promising. They all see actually immediate response in all the patients. After a couple of hours, they report they feel much better. They let's say they come for the injection in a wheelchair, and after a couple of hours, they don't need the wheelchair anymore. They feel less pain and everything which is related to the clinical performance and subjective performance of the patients. This is really good news. And this is true for all the patients. But what my colleagues from India told me that this first effect seems to be more pronounced in the case of actinium-225 injections. So this has nothing to do with a long-term effect for tumor shrinking and so on. But this is a kind of non-systematic observation. I would just say it's encouraging, and it's worth to really have a look on it.
Definitely. Yeah, thank you, Professor. So we have one participant that raised a hand. That is Dr. Hendricks, I think. So please, you can ask your question.
Yeah, thank you, Owen. Can you hear me?
Sure,
Yes.
sure.
Okay, thank you. I have a question. In your first presentation, some of the FAP inhibitors, they contain a nitrile group in their structure. Do you think this group important for binding?
Absolutely. This is a warhead. This is a warhead of the inhibitor, and this is essential.
Okay. One thing crossed in my mind, can we radio label the inhibitor with carbon 11?
At what position?
At the nitrile group, the CN.
Yeah, I know what you mean. I know what you mean.
Is this possible?
I think this would require to see what is the synthetic strategy of the molecule. Saying what I want to say is at what time or step of the total synthesis this nitrile group is introduced. But you must arrange the synthesis, the organic synthesis, in a way that this nitrile substitution will be the last step of the overall synthesis. Yeah.
Okay, maybe I have one more question. In terms of their structure, do you think it's possible if we dual label them with radionuclide and fluorescence molecule to make a hybrid? Yeah.
Yeah.
Okay.
This is at this position at the canoline ring, you may substitute basically at each position, not only the one I've shown, but at each position. You will use, according to the modification done by the group in Antwerp, you will lose a bit in affinity or a bit in selectivity, but more or less it works. And instead of the substitution of a linker and the radio chelator part, it is straightforward to introduce a fluorescence moiety.
Okay. Okay. Yep. Thank you.
Yeah, thank you, Dr. Henry. So we are going back to the chat box. It's a really interesting discussion today. So then another question, I think, from Dr. Rin. You want to ask directly, or Dr. Rin also from Germany, from Dresden, graduate from Germany, I mean. So you want to ask directly? Okay. I think I will just read the question. So it is regarding the affinity and selectivity of FAB inhibitors. Is asking about the minimum selectivity ratio for the target to obtain the ideal FAB inhibitors. How to measure that? Do you have any idea about that?
The first question is affinity. So what we now have available are FAB inhibitor monomers, but also dimers with about 1 nanomolar affinity, even less. We have recently reported a new dimer, which is 1 order even better in affinity. They all work more or less the same. So my main concern would be the selectivity, because some of the other protease enzymes are localized throughout the body, so not overexpressed in the tumor microenvironment like the FAB. So if we are losing selectivity, let's say still remaining the high affinity, but losing selectivity, we will see increasing uptake in healthy tissue and other organs. And this is the point which is more important than playing around with a bit more affinity. It is important to keep the extraordinary selectivity to avoid seeing radioactivity in healthy tissue, both for diagnostic and for therapeutic application.
All right. Is there any comment, Dr. Rin, or is it good? Okay.
Thank you.
All right. Thank you. So we go to, we still have really enough time. So all the participants, you can raise your hand. You can discuss here. So we just make this lecture today is fun, and we have a lot of insight. We have another comment from Dr. Arifuddin Ahmad. Is from the hospital. Is the one of the leading hospital for the nuclear medicine in Indonesia. Is Dr. Arifuddin want to ask directly? You can turn on your microphone if you are still here. Okay. Maybe I will try to read the question. So he asked about whether it is possible if we can also participate in developing radiopharmaceuticals using data chelator in Indonesia. It means if we have several cancer targeted compounds. So is it yeah? What do you think about that?
The answer is yes. Of course, we are always looking. We are happy for collaborators, and that's true that we focused on only only 3 targeting vectors, but there are much more, and now it's straightforward to synthesize new compounds based on these chelators. The chelator itself, the data5M, ready for conjugation, is easy to prepare, or it's also possible to ask a company to synthesize it and have a batch of this data5M ready for coupling. So this is straightforward, and yeah.
Yep, great. Thank you. Is Dr. Arifuddin want to give a comment?
Oh,
Yeah, sure.
Professor, yeah, thank you. Nice to hear that.
You have another comment? You have another question? You can go.
Several years ago, we wrote a proposal to build a collaboration with your university, Professor. But unfortunately, pandemic comes and then we cannot continue that. I hope we can collaborate later in the future.
Sure.
But I should mention that I think recently this platform of hybrid chelators was transferred to a big radio pharmacy company, ITM in Munich. They now realized that this is an interesting direction when both developing instant kit type preparations for gallium-68 and lutetium-177.
Thank you, Professor.
Thank you. And here we have one more question, Professor, if you don't mind. So our colleague is asking about still about the first presentation. When you mentioned about the multi-dimer, homodimer, multi-dimer, and is it a similar concept with multivalent or bivalent? What do you think about that?
I'm not sure about what is the definition of bivalent or multivalent. Do you mean bivalent is similar to heterodimer?
Yeah, that is the question. Is it like a similar concept with heterodimer, or when we talk about the multivalent?
Yeah. There almost the last, I would say, decade, I've seen a number of publications exactly describing this approach. They used two different targeting vectors and combined it in different way into the same molecule. Let's say combining an RGD moiety and PSMA inhibitor. There are really excellent, mainly preclinical data to demonstrate that there is an advantage behind this. So we also had this concept to use a bivalent or let's say heterodimer compounds, and we synthesized a molecule which contains the PSMA inhibitor part and a bisphosphonate. So our concept was that for these late stage prostate cancer patients, the main problem lies in the bone metastasis, which originated from primary cancer. Yep. And if you could apply a heterodimer which both and simultaneously targets the same metastasis by addressing both the PSMA and the hydroxyapatite structure in that bone metastasis, this would increase in an absolute level the accumulated activity and increase radiation dose, and reduce the same time uptake in the glands. So this is what we have demonstrated in vitro. So the bi-specific or heterodimer showed still high binding affinity to PSMA in tumor cell lines, and showed high uptake in binding to bone structures. So the cumulative effect there is important. But that's what I mentioned also for the other studies. This now would need a systematic application in patients, just to verify that all what we know from in vitro and then small animal imaging can really be translated into patients. And I believe that a parallel and simultaneous binding of those constructs will really be helpful.
Yeah, thank you, Professor. So, is there any other question from the participant? So, before we continue, so I just think both of your presentation when we talk about targeting the tumor microenvironment, because my previous project in Glasgow is about tumor microenvironment too. So I just imagine about when we talk about there is tumor microenvironment is I mean it is very complicated. And there is a cold tumor, is a hot tumor that is has a different immune related with the tumor microenvironment. Do you think what is the best approach when we design our molecule? Let's say when you first presentation you targeted like a prostate cancer that is, but how about like lung cancer with or in type of like a colorectal cancer with as a cold tumor? And lung cancer as a hot tumor. Do you think there should be a different approach for that?
No, it's exactly the same. It is exactly.
All right.
The journals are full of, I would say, proof of principle applications or case reports for a number of patients with a number of different cancer. And it appears that the gallium-68 labeled Fabi monomers, they all show uptake and have a clinical value. The question is whether for a particular cancer it is superior to FDG. And there now are the first systematic studies being published demonstrating this comparison. And I know that soon there will be a paper on maybe 100 patients, breast cancer patients, which were traditionally diagnosed with FDG, but also now with the gallium-68 Fabi monomer. And the conclusion would be that the first line diagnosis would be the FAB inhibitor and not the FDG. And so also all these FAB inhibitors show up in almost all the cancers. I think we must be fair enough to say, is there an advantage compared to FDG? And this indeed may depend on individual kinds of cancer.
All right, thank you, Professor. So there is one participant, I think that is Mr. Basit Fabian, raise hand. You can ask your question.
Okay, can you hear me, Professor?
Yes.
Hello. Sorry, I tried to turn on my camera, but it is some problem with my device. Hello, nice to see you in health condition, Prof. Yes, I thank you for your interesting explanation about same way Dr. Wan say about tumor microenvironment and the important or needs to destroy the stroma. Maybe it's still I don't know should we destroy that or not. So how about if we design some radiopharmaceutical which can diffuse inside the cancer, the stroma, with its mechanism? For example, we try to research about the nanoparticle with the radionuclide inside that nanoparticle, and it is maybe the delivery is only with the enhanced permeability and retention. So it is broad tumor habitats rather than tumor specific receptors. Maybe can this strategy has the potential to overcome the tumor heterogeneity problem, and could it be used to design the diagnostic and therapeutic for the cancer? That's my question.
I think so. I think so. But if I can make a suggestion, with these nano structures you are talking about, they do not accumulate specifically in your tumor. So why not to have, let's say, a heterodimer structure, like
Yes,
keeping your nanoparticle intact, but load it with FAP inhibitors. So this then will hopefully result in a, let's say, more selective accumulation, or let's say transport of your full molecule construct to the tumor environment and keep it there, and then hope that once it is there, it diffuses into the orange.
I like that orange orange picture from you.
No, I was thinking about this. It was not a good example because what I learned is the relationship between the shell and the inner part of the fruit is not the one we see in the tumor. It should be something opposite, with a larger shell length and a smaller inner part. I don't know what fruit it may be, but this.
Maybe we have dragon fruit in there.
Yeah, I was thinking about dragon fruit. but this could be an approach to have. In general, I would say I'm talking about drug delivery systems. You could also design, and that's what we do right recently, combine the FAP inhibitor with a cytostatic drug. And there are nice strategies to design chemically those molecules and to guarantee that the binding to the enzyme, the FAP, automatically cleaves the drug, the cytostatic payload. So this could also, it is combining two small molecules. But I recently saw a manuscript on using the same strategy for a monoclonal antibody. But you may think about extending it to a nanocarrier system.
Yes. We also try to apply that strategy to build our radiopharmaceutical research. Thank you, Professor. Thank you for the answer.
Thank you, Mr. Basit, for the question, and thank you, Professor, for the very comprehensive answer. Actually, I think we have a very limited time here. So before we end and I summarize this session, I really want, if you could give us some words. I think currently in Indonesia, our research is mostly for the development of radionuclide reactor based, and our hospital actually still mostly we are using iodine 131, and some we are using samarium for the bone. I think we already discussed for that one with you when we are good minds, and we are do a research about lutetium 177 here, either for PSMA, and we do for the antibody labeling. Maybe you think what kind of research that will be a potential for the further development with our, if you can imagine our current situation in Indonesia.
So this was mentioned earlier, given your situation that you do have lutetium 177 available, and it is good for FAP inhibitors, I would say that could be a good direction, because this reactor-produced lutetium, you would not disturb the performance and the pharmacology of the labeled FAP inhibitors. But this immediately applies to therapeutic compounds. So I guess you would do first steps in that direction, but you always would need to have a PET monomer, FAP monomer beforehand, because it's like always in theranostics. You must screen the patient beforehand to see what the specific uptake is in that particular tumor, and only if there is high enough uptake, then you continue with the lutetium labeled dimer. This is so you would definitely need to have a gallium-68 available. Do you have F18 available?
Yeah, but in some hospital we have a baby cyclotron, so we produce fluorine 18, but in our agency we are ongoing to get the cyclotron, and hopefully we can get that in the near future. But yeah, that's a really nice suggestion from you, I think.
What about gallium-68?
Some hospital using a gallium-68, but not that much. I mean, not we are to be honest, we are not yet goes to the development of gallium-68 currently. But this would be our
you I think you shall identify a hospital which is able to provide the diagnostic monomer and because this is essential to have this in a theranostic combination. Yeah. Did you actually think about producing gallium-68 at cyclotrons?
Sure. This will be our plan, because in our new agency we will try to provide from our local product for all the therapeutic isotopes, especially. Yeah.
It works. It really works.
Hopefully.
We here. I mentioned that now I'm in Berlin, and there's a very good private nuclear medicine department with cyclotron. And within 1 year, they established gallium-68 production, had a very nice solid target system at a baby cyclotron, and they produce 20 gigabecquerel of gallium easily. So, then, but I'm still thinking about if you plan with lutetium Fabi dimers, you always must make sure that the gallium monomer is available.
Yeah. Thank you so much. And I would like to summarize our today's session. And once again, for targeting the tumor microenvironment of FAP using a FAP inhibitors, so a very promising approach, and with a very good result in clinical trial. And there is a lot of strategy we can do, like modifying the linker, and some like introducing the albumin binding, modifying the structure, or using FAPI dimers. And for we will like, when we talk about the radiometals, we need to improve our radiotherapeutic agent with high in vivo stability and good labeling for room temperature. We can do a couple of approaches that, let's say, like using develop the chelator. And once again, thank you very much, Professor Rose, for your time today. This now is almost 6 o'clock in Indonesia. I think it's almost 12 o'clock in Germany. And I know you are still have a lot of things to do, and hopefully you are still well and healthy. And after hopefully I can visit you in Berlin when I go.
Yes.
I think, yeah, sure. I will contact you by email.
We really miss you.
Let's meet in Berlin or Indonesia.
Sure. Hopefully, and I would like to go to the back to the host of this webinar. Ms. Irma,
it was my pleasure. Thank you so very much. Well organized.
Thank you very much. We would like to thank Mr. Ahmad Kurniawan, DVM MSc, and Professor Dr. Frank Roche for bringing us to a very productive session today. So ladies and gentlemen, we are currently in the closing session. We would like to remind you that the link to the attendance record form is now available and active. The link has been embedded in the chat column. Please fill out the form on the link carefully and completely to avoid identity writing errors on the certificate. On the same link, you will also be able to download certificates. Ladies and gentlemen, on behalf of the committee in charge, we would like to express our apologies for the mistakes we made during the event. Thank you for your active participation today and have a great rest of your day. See you later. Wassalamualaikum warahmatullahi wabarakatuh. Thank you, everyone.
Waalaikumsalam
Thank you, Professor.
Thank you,
Have a nice
Professor.
Thank you.
day.
You too. Nice evening.
Yes.